Our next speaker is a colleague, Dr. Takeshi Yoshimatsu, which is a doctor in neuroscience and a research follower in Sussex University. His work focused on the function and the development on the vertebrate and the visual system in zebrafish and mice. He did his PhD degree in the University of Tokyo in Japan, and he received his postdoctoral training at the University of Washington in USA in Dr. Rachel Wong's lab before joining Professor Tom Baden's lab, where he is currently as deputy of the lab, if I'm right. Alongside with his research, he is also actively involved in trends activity as volunteer and also as a startup member of BioRTC. And Professor and Dr. Takeshi, if you are ready, so you could start your talk.
Thank you, Ralph, for the kind introduction.
Yes.
So before I start my talk, I just wanted to briefly mention about how this BioRTC started, because it was kind of like, aligned with the idea that Tom just mentioned, like, what's the original spirit of the trend. So it kind of started in like a couple of years ago. So then in 2019, when Mahmoud actually organized a scientific course in Nigeria at the University of Lagos, and he invited me to be a teacher in the school, the course. And then it was very nice experience to me. And then, we had lots of interaction with the student. And then after we came back from the course, and then we are talking about what to do next. And then we are talking maybe organize a, course somewhere else. But then it occurred to us, like I remember, like, we always sort of like rely on the, getting the funding from charity. But then that's always have a limitation. So then we thought that we should have set up, our own lab. And then also we should then do the actual science that can be published in the high impact and then get the, research money by itself in the way that, this can be sustainable and they could even grow. And then since then, Mahmoud really pushed this idea and then just involved many more people and the higher places also. And it very nice to see this BioRTC launch a symposium today. Thank you for all the effort Mahmoud made. Okay, so then now I would like to start my talk, which is gonna be here. So, okay. Hope you can see it now. All right. So I am working on the visual system and then try to understand how the animal's visual system works. And for today, I'm just gonna focus.
Okay. Can you, the slide we are seeing also shows the next slide. So if you can swap.
Okay, so how do I do that? I don't actually know. Show the display setting, I think. How does it work? Is it
great?
It's good. Okay. Yes. So today's talk, I'm just gonna focus on one of the important function of the retinal circuit, which is about getting the high acuity vision. So what is high acuity vision? Is that it's just being able to see details, like a better spatial resolution. So to explain how the retina works, sometimes it's easier to actually compare with a simple digital camera, right? So how does the spatial resolution look like in the smartphone camera and the human eye? And probably if I show what's the spatial resolution for camera and then compare with human, you might be disappointed to know that even like a simple camera on the smartphone nowadays have 12 million pixels, whereas human eye our eye has merely just a 4.6 million pixel for the color vision, right? So it kind of like sounds like our visual system is worse than the smartphone that you actually have in your hand. However, the Retinal doesn't work as a digital camera. It actually has a specialized region called the fovea, which is indicated as a square in the picture here. And then the zoom in is on the right. So there is this very small area in the retina, which is just have like a 1 or 2 millimeter in diameter. And then there's this dent here. And this region is very specialized. One specialization is that this region has a higher density of photoreceptor, type of photoreceptor called cone photoreceptor. This is basically responsible photoreceptor for our normal daylight vision. And this density is highest in this small area called the fovea. And actually, if you calculate the density and how much area this area is seeing with, basically, this human eye actually concentrate the cone photoreceptor by concentrating the photoreceptor in small area, we achieve the spatial resolution of about 35,000 pixels per square degree, but only in the small area. And this actually, the spatial resolution is much better than the smartphone that we have. And in the smartphone case, because the spatial resolution is uniform across the visual field, so then if you average out, the resolution is 13,000, whereas for the human eye, in this particular region has a much higher spatial resolution. And then the way our vision works is that we use the fovea to scan across the visual field and then reconstruct the higher spatial resolution image. So the way it works is quite different, but it's more efficient. So then, okay, so to get the higher spatial resolution, the one way to do it is to having the area where the cone photoreceptor density is highest, right? But is that the all mechanism to have the high acuity vision? So then to study like whether there's any other mechanism about the high acuity vision, we use the Zebrafish as a model system. So Zebrafish advantage is that we can see through the eye and then we can study the function and so on. Okay, sorry. So then first to look at the visual retina of the Zebrafish, I kind of wanted to show how the visual world look like in the Zebrafish natural habitat. So this is a picture of the Zebrafish natural habitat taken in a river in India. And this picture contains about 1 million pixels. So then it has a quite good resolution and you can see so much detail. But actually the Zebrafish, especially like a larval Zebrafish, does not have 1 million cone photoreceptors. And it actually has only like 3,500 pixels for one color, especially in this case, like a UV cone, okay? So then imagine if you see the larval Zebrafish seeing this image with just a 3,500 pixels, the image looks like this, right? So then if you are a larval Zebrafish, try to capture a small prey, like this object, for UV cones, it would look like this. So it's really difficult to capture the small object for larval Zebrafish. So how do they do that, right? So one way is that having the area where the photoreceptor density is higher, right? So then we map the density of the photoreceptor across the eye and project it onto the visual field of the Zebrafish. So then this magenta map is the front of the larval Zebrafish, and the higher the color is, the density of the UV cones, higher the density of the UV cones, right? So then when Zebrafish converge their eyes, then the upper visual field actually is seen by the highest UV density. In this case, 0.5 cones per square degree. So by having this small area where the higher density of the photoreceptors, zebrafish can improve the spatial resolution in particular region. But this is not only the way to increase the higher spatial resolution. Another way is to having the specialized structure of the photoreceptors. So if you look at this picture, so the dorsal is up, ventral down, and then nasal left, and then AZ is the acute zone, so the high acuity area. So, and if you look at the zooming of the picture, the UV cones in the acute zone has the longest outer segment, which is indicating the green. And then that means that the photoreceptor UV cone in this case has the higher efficiency to capture light, right? So then it has much better signal to noise ratio. So this is one way to increase the signal. Another way, how about the function? So to study the function of the UV cones, we use the genetically encoded sensor. It's called the sensor. And in this case, I'm just showing the picture of the sensor for the glutamate, which is a neurotransmitter released from the cone photoreceptor. And by expressing this sensor in the cells, you can detect the glutamate concentration by the increase of the fluorescence. So for our case, we express this sensor in the postsynaptic cell of the UV cones, right? So that the picture is in the left bottom. And then you can also label the UV cones, in this case in magenta. And then now we can give the visual stimulus, and then we can see how the sensors respond to the different visual stimulus. The visual stimulus we gave is shown in this panel here. Now, by looking at how this photoreceptor respond, and then we can also compare the photoreceptor response in the different regions. So here is the example of the photoreceptors in the acute zone and then dorsal, right? And then these two are the zooming of these two photoreceptors. Here, left is acute zone and right is the dorsal. And then the visual stimulus is exact the same. And then we noticed that actually the photoreceptors in the acute zone respond far more often than the dorsal photoreceptor. So this is quite interesting. Because it's not just the sensitivity, but somehow they carry more information. And then here is the quantification, like how much information they carry, which is done by the computed by the Cornelius in the field parents lab. Right, so then this red line indicates how much information this acute zone photoreceptor can carry, and then it's much higher compared to the rest. And then we wanted to know how this happened, and then we next look at the calcium concentration, because the calcium is basically the key factor for the neuron to evoke the neurotransmitter response. So then we examine the calcium concentration across the eye by looking at the light intensity of the calcium sensor subtracted by the endogenous expression level, sorry, not the endogenous, but the expression level of the sensor. And then we quantify the calcium level concentration in the photoreceptor. And it turns out that the calcium concentration is higher in the acute zone. So what does that mean? Right? So if you have the higher calcium concentration, this means that this photoreceptor can respond to the flash of light in both ways, in the sense that the calcium concentration could go higher or even lower. Whereas if you have the calcium concentration lower, you can only increase the calcium concentration, but they cannot go even lower. So that means that this acute zone can have twice as much information than this dorsal photoreceptor. Although this is not quite twice as much because the response to this dark flash is much stronger in dorsal. So then it's not quite twice as much, but still this acute zone photoreceptor can have more information, right? And then this, we think, underlie the higher information rate from the glutamate response, right? So if you look at the trace in the calcium from calcium or glutamate, and then this gray bar indicates visual stimulus, then the acute zone UV cones, calcium has far more calcium response compared to the UV cones outside the acute zone. And then this kind of corresponds to the glutamate response. Okay, so just to summarize, there are several mechanisms to support the high acuity vision in the very specific area. One is that having the higher resolution. The other one is that the encode the high the contrast better. And then as a result, so if the photoreceptor is uniform and then physiologically uniform, rubber zebrafish will not see the prey here, but by having the special vision, they now could see the small object right here. So using the zebrafish, we could sort of like, we found that there is sort of like, not just a concentration, but there are other mechanisms to enhance the high vision. But as you can see, the retinal structure, if you compare the retinal structure in the Fovea in the human and the zebrafish acute zone, it's not quite the same. But then there are many other animals that actually have this retinal structure that quite resemble to the human Fovea, such as like a seahorse or hawk, also has a beautiful Fovea-like structure. And then even some species Anolis has nice deep one. And then also, this is one of the gecko, dwarf gecko, which is very common in Africa, has unique structure of the retina. And as Tom mentioned in the previous talk, that by comparing this specialization, and if you closely look at this retinal specialization, each species has different kind of specialization. And then by looking at why it happens in this species, and then by looking at the environment they live, we could probably understand why each of the structure uniquely serve to their specialized environment. With that, I'd like to just acknowledge the people who helped, and then also the collaborators. And then thank you for your attention.